we are looking at, we are still trying to understand the physiology of stomach. Okay, stomach, hello, physiology of stomach. Okay, and then we had in the previous class we focused on a very interesting aspect, that was a parietal cell. You know, parietal cell. Parietal cell. What is the? Yeah, it's cell secreting. Very good. Okay, and then we have seen actually how the cell secretes hydrochloric acid, and then we have. So we'll carry on now. So let us look at this diagram. We saw it also last time. You can see the esophagus is coming that way. Okay, and then you have the fundus part, and the lower part that is towards the duodenum will be the antrum part. Okay, so far, good. And in the fundus part, we have those. The author is showing us two cells. Okay, both of them are parietal cells, and both of them are capable of secreting what and what? HCl and intrinsic factor. So thousands of cells they are. So the same cell can secrete the two substances. We'll talk about it each. And then there are deep into the gastric pit. Deep into the gastric pit, we have chief cells. Okay, which are secreting the most important protein digesting enzyme, that is pepsin, that is also in the stomach. And then the author draws our attention to very interesting group of cells which are in the antrum part. Now I am talking about it. Okay, and we call them as G cells. What do you call them as? We call them as G cells because they secrete a peptide hormone, 28 amino acids. How many amino acids? What is the name? Gastrin, G-A-S-T-R-M. Name indicates that it is coming from stomach. Okay, I-N at the end indicates that it is a protein. Okay. So G cells secrete a peptide hormone which is called as what? Gastrin. Okay, and guess the very interesting. Stomach is an endocrine gland. We made this point in the previous class, and let's see now what gastrin essentially does. Amazing. Oh, this is just to tell you it's a peptide. Okay, and has a certain sequence of amino acids. But now what gastrin? Now this is a very interesting diagram, and I want you to focus and understand. You have to understand this. You can't run away from this. Beautiful diagram. Beautiful diagram. We are looking at the stomach. Very clear. Very clear. We are looking at the stomach. In the stomach, then, as we have said, as we have observed in the previous one, if we focus on the fundus part, and there are cells, we have already seen that those cells are responsible, they are parietal cells and they are secreting acid and intrinsic factor. We are looking at the same thing. So, this is the cell here, the black dot represents the cell. What cell it is? What cell? Parietal cell. What cell? Be in the class. What cell? Parietal cell. Very good. This is the parietal cell, and for our convenience, the author has taken this square here and blows it up here, and here is our parietal cell. And parietal cell is secreting HCl and good. Now, this parietal cell, on its basal side, author has clearly shown 3 different types of receptors. How many types? We have to remember each. 3 different types of receptors, and stimulated by any one of these receptors, the cell gets highly excited and they start pumping hydrochloric. So tell me, so how many factors are there that will control the release of hydrochloric acid by the parietal cell? 3. Okay, and the 3 agents are: number 1, very simple, one is acetylcholine. One is what? Acetylcholine. Now, where is this acetylcholine coming from? Very simple, this acetylcholine could be coming from the vagus, which means parasympathetic nervous system. Okay, preganglionic, postganglionic. It could either directly go, or it could go on the neurons of the, remember enteric nervous system. Okay, but then whatever it is still acetylcholine, and the cell is getting acetylcholine, and this acetylcholine is acting via a receptor. What kind of receptor would you find? What kind of acetylcholinergic receptor will you find on the basal side of the parietal cell, basal side? M3. What time? So muscarinic 3 kind of, and muscarinic 3 is that receptor is also 7, it's a 7 transmembrane, okay, G protein coupled system, and when excited it will excite the cell to generate hydrochloric acid. One stimulation. The second stimulation is H2. What does H represent here? Histamine. Histamine comes from a different type of cell which is sitting in the vicinity, very close, next there, sitting right in the neighborhood is another cell. What do we call it as? H cell is the source for what? Histamine. Say that loudly. We have seen the structure of histamine in the previous one. Okay, and histamine, do we call histamine as a hormone? No, we don't call it as a hormone. Okay, some people also call it as a local hormone. Okay, or an autocoid. Means what? Histamine will be released, not in the blood, in the vicinity, and once it is in the vicinity, it will travel around, and it can travel generally it can travel half mm or 1 mm. What happens that then after that there are enough enzymes in the extracellular which will chew it up. So histamine release will act in the neighborhood. So within the circle, okay, within a sphere of about 1 mm on all the sides, what all the parietal cells are there, they would have receptors for histamine, and they would be stimulated. And histamine receptor, what kind of histamine? Histamine receptors also two kinds, H2 type, which again it is 7-transmembrane G protein coupled system. What am I talking about? Histamine as a signaling molecule coming from what kind of cells here? Enterochromaffin. Say that loudly. So enterochromaffin cell, which is also sitting in the neighborhood in the stomach, okay, can release, and that can, okay. And then there is third kind of receptor which is called as CCKB or CCK2, whatever. Now CCK stands for cholecystokinin. Say that again. Cholecystokinin. This funny name comes from this is interesting. Cholecystokinin. Cholecystokinin is itself a peptide hormone. What is it? It's a peptide hormone. Okay. Now the moment you say it's a peptide hormone, you should ask a question, what is the source? Okay. And its abundant source are the cells in the duodenum. Cells where in the duodenum. Are we talking of duodenum? We are not there yet. We are still in the stomach. Okay. I'm giving you a broader picture. So if you just fill in the blanks, cholecystine comes from where? It comes from certain cells in the duodenum. What is it? It's a peptide hormone. What does it do? Basic function. Its function is to get released in the duodenum. It's a hormone, get into the blood, travel with the blood as far as the liver and as far as the gallbladder. Okay. Gallbladder contains lot of substances, no enzymes, but one of the very important system is cholesterol. Cholecystokinin is cholecystokinin, cholesterol. So kinin, cholecystokinin. Cyst is a bladder. Chol contains cholesterol. Cyst is a bladder. Kinin is to mobilize. Cholecystokinin. Okay. So what is cholecystokinin? It's a hormone. Where will it go and act? It will go and act on the smooth muscles of the gallbladder, which will have receptors for cholecystokinin, and under that influence it will what? The gallbladder will do what? Contract, and it will expel what? No, it will bile. It will bile, okay, which will contain cholesterol. It will release what? Bile. Where will the bile go? Bile will go into the duodenum and it will do its function. What function? I will tell you eventually. Okay. Have you observed this point? Now, here the story gets a little bizarre. We got cholecystokinin, we got its receptor earlier in the history of science. We got it. So we knew earlier, 1960s, 70s, we got cholecystokinin, we got its function. Got the receptor, okay, we call it cholecystokinin receptor. Okay. So far, are you okay so far? Okay. Now we find that gastrin, which comes from the endocrine cells in the antrum part, which we have seen in the previous slide, can act on the same receptor. So therefore, instead of calling it as a gastrin receptor, what do we call it as? CCK receptor, cholecystokinin. We didn't change the name. So whenever you look at this image, you may be slightly confused as to with what? Well, I'll tell you. Now, keep the confusion in your brain, I'll sort it out within 2 minutes. Just as we have seen this cell here, I'll draw your attention to yet another cell with which I spoke to shortly, and this cell is nothing but a gastrin cell. We have already seen gastrin is a peptide. Okay, it is in the antrum part, and it's a hormone, so it gets into the blood. So here you will understand the meaning of this gastrin cell is being released by this cell. Okay, and where does the gastrin go through the blood? It goes to parietal cell. And where does it act on the parietal cell? This is the same thing, this is the parietal cell, and on the parietal cell there is a receptor. That receptor is also 7-transmembrane receptor. Somewhere outside it can bind with gastrin. It can bind with what? Gastrin. It can also bind with cholecystokinin. It can also bind with cholecystokinin. Okay, that's why the name. That's why the name. But here it is combining with cholecystokinin. Life is never simple. Two types of receptors: cholecystokinin A, cholecystokinin B. Some books also called as cholecystokinin 1, cholecystokinin 2. But cholecystokinin 2 is same as B, A is same as 1. Simple. Okay. Hello. Done. Great. Okay. So now what author has done for convenience is you pull out a cell from here and blow it up here. Good so far? And then author tells us that this is a G cell, and what does it secrete? Gastrin, and it comes into the blood and goes into the, goes like this here, comes here like this, and acts on its receptor, CCKB receptor. What receptor? And whenever it gets information from CCKB receptor, so parietal cell stimulated with acetylcholine, okay, go ahead. Stimulated with M3 acetylcholine, yes, go ahead. I'm sorry, see, this is what and what, histamine, acetylcholine via M3, histamine via H2, and CCK and gastrin via CCKB. These are all the 3 stimulants. Okay. Now let us go to this original diagram and try to understand. You take in food, you are starving, okay, none of this is stimulated, you don't need to. So you have food. Once you have the food has arrived, and the arrived food here can directly stimulate. Let's see, it can directly stimulate the parietal cell. It can, some of it can go down, it can stimulate the gastrin cell, okay, and then the gastrin cell will further, okay. Never forget homeostasis. Okay, you have stimulated parietal cells, and the parietal cells are have become active and generate H ions. As a result of the pH falls. You need to stop somewhere. Are you with me? You got to stop somewhere. So you need to decide that okay, I want so much acid, but not more than that. How do you achieve that? For that, the author shows that once the pH, the parietal cells which are releasing hydrochloric acid, once the pH goes lesser than 3, then they start inhibiting the gastrin cells. Are you with me? So whatever is the protein content in your diet, listen to this, whereas the protein content in your diet will stimulate the gastrin cells to release gastrin, which will go up into the fundus part and stimulate the parietal cells to release hydrochloric acid. More, stop. That stop comes from the acid itself. So in this area, so you can ask a question. I don't know the answer, but I ask a question. Do the gastrin cells, okay, have sensors for pH? Okay, and once the pH goes a particular limit, okay, the gastrin cell itself is inhibited so that it no more sends the signal to the parietal cell. Got the point? I'm not saying I'm telling you that it is. I'm just asking you to think about it. Good. Now, let's see. So what are the 3 stimulants? Again, acetylcholine, histamine. Okay, good. Now, there is a 4th character. Okay, and the 4th character is called as somatostatin. What do you call it as? Somatostatin. Say that again. Somatostatin. Say that again. Somatostatin. Now, somatostatin is an excellent example of the same peptide doing absolutely different things in different areas. Hello, one signaling a molecule. What is it? Somatostatin. What is it? Somatostatin. Okay, but let us see let's see what it does. First of all, the cells which secrete somatostatin, okay, I'll take the story from a different point of view. Let's go to pancreas. In the pancreas, we have islets of Langerhans. Done. Islets of Langerhans. In the islets of Langerhans, there are beta cells. They are source for what? You have done it, Nixon has dealt with it, beta cells. Then there are alpha cells, okay. Then there are delta cells. What are they? There are delta cells. Delta cells are also called as D cells, and they are a source for a hormone, somatostatin. What do you call it as? Somatotropin. So somehow that terminology which we started at the level of pancreas, where you have alpha, beta and delta, delta is same as D, that terminology we have taken here, and here these somatostatin secreting cells, therefore we call as what? Got the answer? Why D cell is D cell? Why D cell is because it is secreting somatostatin. Now why somato should be S cell? Why D cell? Because we borrowed it from. And it also, we also get an interesting lesson here, that somatostatin is a peptide. It is coming from it is coming from the islets of Langerhans. It has one particular set of function. Here somatostatin has what? Okay. Here is a D cell here. Where is D cell? The D cell is here. You are getting it here. In which was fundus or antrum? Fundus. In fundus, are you getting a D cell in the fundus? Yes. This D cell, when stimulated, okay, how is it being stimulated? Again via D vagus, okay, but not directly, or actually it is. I wish I could have something, just transfer the information there, that would make my life simple. Okay, never mind. Okay, so you have okay, can you see the vagus nerve there, preganglionic, postganglionic? The postganglionic nerves will secrete again acetylcholine. Yeah, okay. It is secreted along with yet another peptide, okay, which is called as calcitonin gene related peptide called CGRP. Biology can be very funny. So what am I doing? I am bombarding with a lot of peptides, okay. So I am telling you one more peptide. You see, you got it. I mean, you came here, okay. So you have to suffer. So what is the new peptide I am talking about now? Calcitonin gene related peptide. Everybody? Again? That peptide is being co-secreted by that neuron along with acetylcholine. That CGRP talks to the D cells, okay, and then D cells release somatostatin, which, look at the red color of the arrow, which means what? Inhibitory. It inhibited cell. If it's inhibited cell, then this axis is stopped and the stimulation of the pancreatic, I made a mistake, of the parietal cell is retarded. Okay. So when you want the parietal cell to secrete, you have so many avenues. When you want to stop it, okay, then via the D cells, okay, you have an inhibitory path by which you can control the. Okay, I can see the pH goes less than 3. Okay, it stimulates the D cells. Okay, the D cells secrete somatostatin. Okay, it is called as SIH, somatotropin inhibitory factor. One more, but it is same. It is what? Somatostatin, and somatostatin receptor also there on the G cell, but look at the red arrow. So somatostatin is doing two functions. Here it is inhibiting the G cells, and here it is inhibiting the H cells. Okay, and both ways, by both the arms, it is ultimately inhibiting the secretion of the parietal cells and inhibiting the release of hydrochloric acid. Are we okay so far? I would urge you to go home and just keep on looking at this diagram, just keep on looking at this, and do it today. Today, when this talk is still fresh in your mind, spend half an hour, just keep on looking. It's beauty of this. If you ask me what is the beauty of physiology, it's here. Amazing. You see how and the review from which I got, he says this is a highly simplified version. Okay, I said okay, I am happy with it, I do not want any more. So this is what, okay, all right, let us move on. Okay, so this is the summary of what we have done. We are looking at the acid secreting parietal cell. The author has made our life simple. Gastrin, histamine, acetylcholine, stimulate, somatostatin inhibit, and here is a list of the factors which will stimulate which will bring about the stimulation of the parietal cell and bring about the, okay. I spoke to you about this point in one of the previous lectures, and what we said, there are In the earlier days in 90s, so 1990s to almost 2000, what was the tool available for the treatment of gastric acidity? Cimetidine. What was it? Cimetidine. It's there in bracket. Okay. And how did it work? It was a histamine H2 receptor blocker, and so histamine won't come, so you will have, okay. However, we had a wonderful magical drug, almost amazing drug which has come up in last 20 years. It's freely available in pharmacy. It is one of the most widely used drugs, and those drugs, that drug falls into what you call as, say that again, PPI. What does PPI stand for? Proton. Say that again. Proton pump inhibitors. Okay. I mean, here in the earlier one, we are going to the histamine and then going to the parietal cell. Here the proton, we are just inhibiting the release of the H ions. Am I with me? Are you with me? So how that happens is these drugs are among the most widely sold drugs across the world. Okay. So one of the most commonly used drug available in the market is called as Omeprazole. What do you call it as? Omeprazole. What is it called as? Omeprazole. And and let us see now how the Omeprazole works. And I got a very nice slide in this paper in Nature. This is Nature Review Drug Discovery, a beautiful slide. Just appreciate the slide. Look at the beauty of the diagram. Author is drawn in front of us a single parietal cell. Comfortable so far? On the basal side, the author is drawing the 3 receptors. We are okay so far. What are the 3 receptors? We are very familiar. This is gastrin, okay, and that is what? That is acetylcholine. And what is that? That is histamine, and they can be inhibited by the histamine blocker there. Can you see that blocker there? And this can be inhibited by an acetylcholine blocker, okay. And here and all the 3 of them can stimulate, and then this cell is stimulated, and this is the on the apical side, the author has shown a single pump. There are millions of them, okay. And once the pump is activated, you have a release of acid. We have done this. Now, you take a tablet of omeprazole. Now, omeprazole molecule has no charge on it, okay, because there is no charge. It's good, it goes everywhere in the body. It goes everywhere, of course. You can't selectively say that go only to the parietal cell. It goes everywhere. Out of the whatever molecules happen to go into the parietal cell just by diffusion, okay, it can easily cross the plasma membrane because there is literally, it's a small molecule without any charge. It goes everywhere. Once it goes into the parietal cell, because of the high pH within the cell, see, there is a magic of drug discovery. You really have to appreciate the beauty. Because of the high acidity within the parietal cell, the molecule omeprazole gets protonated. Means what? It gets one H. I will draw your attention to the omeprazole molecule that we have seen in the previous slide. That is the molecule EAT as a drug. Are you with me? Now, somewhere on this position, can you see there is a tiny N there? Hello, that tiny, can you see that? Can you see here? It is NH plus. Means at that site it is what? Protonated. Then it goes into what the chemistry people can tell you better. It goes through 3 steps, and this molecule is converted into more or less this molecule. This all is happening in the parietal cell. Okay, I am not going to tell you about the chemistry of those because I myself do not understand. But if you go into the literature, each and every step is been fully described. Are you with me so far? Now, once you get this molecule in this cell, this molecule can combine, one molecule can combine with one proton pump molecule. So, not sodium, hydrogen, potassium ATPase, which is your pump for H ions, one molecule combined with one molecule, okay, and the molecule is ineffective. You have killed the molecule. So, how many molecules of HK ATPase you can kill? As many molecules of you have provided. How many molecules of proton? I am sorry. As many molecules of omeprazole modified like this you have provided? One molecule killed. Did you get the story here? Hello. That's it. Okay, so it's a very powerful, and it is irreversible as described by the active form will covalently and irreversibly bind to the gastric proton pump, deactivating the pump. So you won't have any more hydrochloric acid. Yeah, sure.
So yesterday you said that these cells are protected by a mucus layer. It
Yes.
has a pH of 7.
Yes.
But then won't the molecule be deprotonated when it comes near the parietal cells? Because
The answer to that question lies in the that's a very interesting question, and people have toiled with it for a very long time. The answer to that question is here. See, this is here you will get a very thick layer where the pH is 7. The layer here is relatively very thin, and what we talked just now, all that drama is happening here deep inside. Okay, and the omeprazole is getting through the blood. Okay, oh no, omeprazole, you see, omeprazole you eat, gets absorbed, gets into the blood, goes everywhere. It doesn't. No, it doesn't happen. No, it's good you asked. It doesn't happen that way. It has to be absorbed, go into the way, it will pass through liver, everywhere, whatever it, and then whatever it happens to reach into the parietal cell, it will show its action.